Our final speaker of the morning session is Professor Sakaguchi, 2015 Canada Gairdner International Laureate, Distinguished Professor and Vice Director, Laboratory of Experimental Immunology, WPI Immunology Frontier Research Center at Osaka University in Japan. Professor Sakaguchi was awarded for his discovery of regulatory T-cells, characterization of their role in immunity, and application to the treatment of autoimmune diseases and cancer. To stay healthy, our immune system needs to discriminate between itself and invaders or non-self. When unable to differentiate between the two, the immune system destroys healthy cells or tissues, causing autoimmune diseases. Professor Sakaguchi's discovered regulatory C cells or Treg cells, which help maintain order in the immune system between self and non-self. Within his laboratory, Dr. Sakaguchi has demonstrated that increasing the number of Treg cells can prevent and treat autoimmune diseases. Further, Treg cells suppress the immune system against cancer, and there are now several clinical trials in this area. Please join me in welcoming Professor Sakaguchi for his lecture entitled Regulatory T Cells, a Key Controller of Immune Responses.
Thank you, Dr. Yang, for a kind introduction. So today I'd like to talk about regulatory T cells, a subpopulation of lymphocytes. And then, I'd like to talk about how we discovered this population of lymphocytes, and then how it works, and then how it can be targeted to control immune responses in humans, in particular in cancer immunotherapy settings. For many years, my interest is in the immunological self-tolerance, why the immune system it can react with the bacteria or viruses, but it doesn't react with or attack cells or tissues. It is called immunological self-tolerance. If we understand the mechanism of immunological self-tolerance, we may be able to understand how autoimmune disease develops, how allergy develops, or how we can evoke a good tumor immunity against cancer, which is kind of self-like cells. And also, we may understand how persistent infection, like hepatitis B, hepatitis C viruses, how it is persistent, not eliminated. Or ultimate goal of organ transplantation, it is to make the immune system look at others' organs as if it is self. And also long-standing the mystery in immunology is how fetal-maternal tolerance is maintained. So let me talk about autoimmune disease. This cartoon shows that at least 5% of the population is afflicted with some sort of autoimmune diseases. It includes various diseases, organ-specific and systemic. For example, in the nervous system, multiple sclerosis, thyroid gland, Hashimoto's thyroiditis, stomach, autoimmune gastritis, or adrenal gland Addison's disease, and type 1 diabetes, and also systemic ones like rheumatoid arthritis. So the mechanism and cause of these diseases, still it is controversial. But there is one characteristic of these autoimmune diseases, especially organ-specific ones, that is overlapping. So here, patient, he or she develops type 1 diabetes, and he or she may also have thyroiditis or autoimmune gastritis or adrenalitis. So this kind of overlapping is also common in animals. For example, NOD mice, it's a good model of type 1 diabetes. When they become aged, some of them develop a thyroiditis. In BB rats, which was developed in Canada, it's a good model of, again, it's a good model of type 1 diabetes. But at the same time, they may develop a thyroiditis and gastritis. So these findings in humans and animals indicate that the cause and mechanism of autoimmune disease is not organ-specific, there may be it is a kind of there may be a common and general mechanism for these diseases rather than specific etiology for each. So that is our interest for many years. To address this question, there are two good experimental systems. One is neonatal thymectomy of mice, just normal mice. When they are thymectomized, removed of the thymus around day 3 after birth, and some of them spontaneously develop autoimmune gastritis, ovulitis. This is an autoimmune disease of ovaries and thyroiditis and so on. So this was first found, this finding was made by two Japanese pathologists, Nishizuka and Sakakura, late 1960s. So they are endocrinologists. So they thought that maybe thymus is producing a sort of hormones targeting ovaries and other tissues. But it turned out to be autoimmune, immunological. Another good system is using normal rats, and you remove the thymus around 6 weeks of age, and then every 2 weeks, if you irradiate them 200 Röntgen, 2 gray, then some of them spontaneously developed autoimmune thyroiditis and type 1 diabetes. So just removing the thymus or X-irradiation, that's enough to cause autoimmune diseases, which are very similar to the human counterpart. So this is the neonatal thymectomy system. So normal bulb C or A or C57 black mice. They are normal mice, no autoimmunity. And if you remove the thymus around day 3, 30% developed gastritis and 30% ovulitis. But at day 0 thymectomy, within 24 hours after birth, and almost nothing happened. At day 7 thymectomy, and nothing happened. If you change the strain to A, then 90% developed ovulitis together with the low incidence, but the thyroiditis and gastritis or orchitis. However, B6 mice, C57 black mice, no disease. So day of thymectomy is critical, and also genetic background of the mice. That's also important. And the important finding, we think it's important, we made many years ago, is that if you inoculate normal T cells from non-thymectomized adult mice, just 5 million T cells, before thymectomy, or 1 week after, or 2 weeks after, now you can completely prevent the disease development. So this means that normal T cells from normal mice, it contains T cells which has kind of suppressive or autoimmune preventive activity. So this is a hypothesis. So thymus producing T cells from gestational days on, day 0 they are born, and then if you remove the thymus around day 3, now you can induce autoimmunity. So this means that before thymectomy, potentially dangerous, hazardous, self-reactive T cells already migrated to the periphery. And then the regulatory T cells or autoimmune preventive T cells, they may start to migrate around day 3. So NTX the regulatory T cells, and then those T cells which have already peripheralized, they now, in the absence of regulatory T cells, they can be spontaneously activated and cause autoimmunity. So, and after NTX, if you inoculate normal T cells, including regulatory T cells, you can prevent the disease. Now the critical experiment is how we can differentiate between regulatory T cells and autoimmune T cells, because normal individuals, you are not thymectomized mice. And this means that regulatory T cells dominantly control the autoimmune T cells. So the experiment we conducted many years ago, that was if you take normal mice, say bulb C mice, in the spleen, it may include potentially dangerous autoimmune T cells and regulatory T cells. So how we can remove the regulatory population? So we using cell surface markers, we remove the certain cell population and then transfer the remaining cells into T-cell deficient mice. In this case, nude mice, congenitally devoid of thymus. So they have no T cells, but then they do have B cells and antigen-presenting cells. And then we found that if you remove CD5 high cells, then if you transfer the remaining cells into nude mice, they spontaneously developed the various autoimmune diseases very similar to what we see in humans, and plus inflammatory bowel disease. And 5 years later, a group in Oxford, they showed that another marker, CD40RC, is a good marker to induce this kind of disease. And we wondered then, what is a more specific marker for this population? And we came up with CD25. So CD25 positive cells, 10% of CD4 positive cells, and then less than 1% of CD8. So just removing the 10% of the T cells, now you can induce various autoimmune diseases. So here, autoimmune gastritis, and then they have autoantibodies against parietal cells, that is hydrogen potassium ATPase to produce hydrogen ion. And thyroiditis, and with anti-thyroglobulin autoantibodies, and type 1 diabetes. Here, Langerhans islets, and they have inflammation, and the other islets already destroyed. So this particular mouse developed type 1 diabetes. So based on these findings, now we understand that the normal thymus, the majority of self-reactive T cells are deleted, reactive with abundant, ubiquitous self-antigens. But then still, thymus can produce the potentially autoimmune T cells capable of causing autoimmune disease. And at the same time, the thymus produces regulatory T cells, which are specialized for immune suppression. So they are already function competent in suppression in the thymus. So it may take time for them to become mature in the thymus. So there is time difference. That is the reason why the day 3 thymectomy can cause autoimmunity. So if you ask them what's the cause of autoimmunity, whatever it is, genetic anomalies or environmental insults, if they can affect the balance between regulatory T cells and autoimmune T cells, they can be a cause of autoimmunity. So ionizing radiation, already I've told you that in the rat, if you, after thymectomy, if you irradiate the split dose of irradiation, it can cause autoimmunity. And that is because Tregs are highly proliferative by recognizing self-antigen or commensal microbes. And cyclosporin A or MTLV, these biological or chemical substance can cause autoimmunity by affecting the balance. And then you may ask then, is there any direct evidence in humans in which the regulatory T cell anomaly is responsible for autoimmune disease? That is Foxp3 gene mutation. So there is a disease called IPEX syndrome, immune dysregulation, polyendocrinopathy. So many endocrine organs are affected by autoimmune attacks, and enteropathy, inflammatory bowel disease, and disease gene is on X chromosome. So more than 80% of patients develop type 1 diabetes within 3 years after birth, more than 70% thyroiditis, and almost 100% inflammatory bowel disease, and allergy, including food allergy. So they cannot take food orally. There is a mouse strain called scurvy mice. Similarly, they show systemic inflammation, and disease gene is on X chromosome. Around 2000, the gene, responsible gene, was positionally cloned and designated as FoxP3, a transcription factor. And immediately, it was found that this gene was responsible for IPEX syndrome. So we got interested in this gene, FoxP3, because these pictures are very similar to what we had seen by removing regulatory T cells. So indeed, the FoxP3 is expressed by 10% of CD4 positive cells. No such cells in scurvy mice. So if you co-stain the splenic CD4 T cells for CD25 and FoxP3, the majority of FoxP3 positive cells are CD25 positive. So this means that we remove the CD25 positive cells and could induce autoimmunity. So this means that we remove the majority of FoxP3 positive cells. So now the FoxP3 is so far the best molecular marker for regulatory T cells in mice and also humans. So let me summarize this part of my talk. Normal immune system, it harbors potentially hazardous self-reactive T cells. And then, of course, they have the T cell react with bacteria or viruses. And self-reactive T cells, their activation is one aspect controlled by FoxP3 positive regulatory T cells. So if you remove FoxP3 positive cells or reduce their number, certain common autoimmune diseases develop, like type 1 diabetes or thyroiditis. And also the magnitude against commensal bacteria, it increases. So you have the inflammatory bowel disease due to excessive immune reactions against commensal microbes in the gut. And you can also have an allergy to innocuous environmental substance. And so the IPEX syndrome, allergy, immunopathology, and autoimmunity all develop in these patients. So this also means that the self and non-self is not so discrete. By changing this level threshold, you can elicit the certain self antigens, especially cancer antigens. So it's good for cancer immunity by reducing, lowering this level, or by increasing this level, upregulating this level, you can induce a suppression and perhaps a tolerance to organ graft. So this is the experiment for tumor immunity. Again, they are nude mice, T-cell deficient. If you transfer a Treg deficient cell suspension, here these are Tregs. And then if you inoculate the cancer cells, the cancer become bigger and then rejected. After rejection, if you inoculate 10 times the number of cancer cells, immediately rejected. So this means that these mice, they became immune to these cancer cells. But if you transfer 4 T cells, regulatory T cells suppress immune responses, so you see a tumor progression. All the mice died of tumor progression. And if you co-transfer 1-to-1 ratio, now Treg become half in number, but still they are suppressive, and so the tumor cannot be eliminated. For transplantation, they are nude mice, and so easily you can transplant allogeneic skin graft and no rejection. And then if you inoculate normal T cells, then 100 acceptance, and now they are start to reject, and within a month, all grafts are rejected. However, if you first transfer regulatory T cells, then these cells are recruited to the graft sites and then proliferate in an antigen-specific fashion within a week. So after 1 week, if you inoculate normal T cells, now the rejection is prolonged, and then 30% become permanently tolerant. If you increase the number to raise the threshold of the immune suppression, now the 70% become permanently tolerant. So without immunosuppressive drug, just on targeting the naturally present regulatory T cells, you can induce a stable graft tolerance. So now we understand that the thymus produces FoxP3 positive regulatory T cells. They are already functional in the thymus, and anomaly of this population can be a causative of autoimmune disease or allergy or IBD. And also by targeting them, you can induce tumor immunity or establish transplantation tolerance. So just one slide, how they suppress the immune responses. The most important function of FoxP3 is, of course, FoxP3 transcription complex. It controls hundreds of genes. The important target is, one is IL-2. So FoxP3 replaces IL-2 production. So Tregs are dependent on exogenous IL-2. So without IL-2, they cannot survive. And also another the function is to keep the expression of CTLA-4, a co-inhibitory molecule, high level. And then these CTLA-4 down-regulate CD86, this is a co-stimulatory molecule expressed by APCs. So responder T cells, they are deprived of IL-2 and also deprived of co-stimulation. So depending on TCR affinity for antigen, they may become apoptotic or anergic or just stay dormant. Now let me move on to how the regulatory T cell function is maintained. So as I told you, they are produced by the thymus, and their task is they must go to the inflammation site and suppress the immune response. So inflammation sites, there are lots of pro-inflammatory cytokines. So if they lose the function and become normal T cells, they are dangerous because now they are involved in the attacking the target tissue. So how their function is maintained, that is a question. And also recently, the FoxP3 positive cells, they can be produced from naive T cells, at least in vitro, in the presence of TGF beta and antigen and IL-2. So we call the natural Treg produced by the thymus, and also induced Treg produced from naive T cells. So just let me give you an example of why the stability of Tregs are important. So natural Treg expressing FoxP3 and in vitro induced Treg similarly expressing FoxP3. And if you remove the Treg and if you transfer them into lung minus mice, T cell B cell deficient, you have colitis. And if you co-transfer with iTreg or nTreg, which one is stable? So here the black line, just without Treg, these mice start to die by systemic inflammation and colitis. And then natural Treg, blue line, completely prevents the disease. But the iTreg, red line, they are not so effective. Why? This is because their expression of Treg-associated molecules, like FoxP3, is not so stable. So after transfer, soon they lose to the expression. CTLA-4, Helios, and CD25. These expression is now they are losing their expression. So how the stability is maintained? So the most, of course, this is epigenetics, and the most important one is DNA methylation, because DNA methylation or demethylation, this is highly heritable along cell division. It is relatively stable. It is linked with gene expression. For example, methylated CpG has good correlation with gene depression. On the other hand, the demethylation has good correlation with the permissive gene expression. So first we looked at where the Treg-specific the demethylation or methylation. Here, this is a MADIP sequencing, DNA chopped down to a small fragment and immunoprecipitated with anti-methyl cytosine antibody, and we did a deep sequencing. So now you see the Treg versus non-Treg, naive T cells, this kind of peak you see. And here, this peak, it is specific for naive T cells. And then in Treg, demethylated, not methylated. So these regions in the genome, among 160,000 the methylated regions, only 300 they are specific for Treg, 0.19% of the genome. More precisely, by sulfide sequencing, you can more precisely locate where the demethylation is. So here, FoxP3 gene, this is a peak in conventional T cells. This corresponds to here. So each strip corresponds to each CpG residue. So normal T cells, methylated, blue, and then Treg, demethylated, yellow. Similarly, EOS here, CTLA-4 here, and CD25 here. So among 300 regions, we chose 5 regions as Treg-specific the marker, epigenetic marker. So CD25 are a bit less stringent, but still normal T cells, whatever you stimulate them, no change, but then natural Treg show very stable demethylation of the Treg specific demethylation site. So TGF-I Treg, no change. FoxP3 transduction by retrovirus, no change. Or memory cells or other T helper subset, no change. So this Treg-specific epigenetic pattern is highly specific for natural Treg in mice and humans. And this is ontogeny. So Treg produced in the thymus, already in the thymus, they show the FoxP3 positive cells show Treg-type epigenetic changes. And this is kinetics. Gradually, they have more stable Treg-type epigenetic changes. And is this FoxP3 dependent or not? So we made a mice showing expressing GFP when developing T cell receive the FoxP3 inducing signal. So here this is FoxP3 protein staining. So normal T cell, normal mice, the Tregs are FoxP3 protein positive and also GFP positive. But here Treg scurvy mice. So endogenous FoxP3 gene has mutation, so no expression. But when they receive the FoxP3 expressing signal, GFP is on. So we call them FoxP3 null Treg. These FoxP3 null Treg, they show similar the epigenetic changes already in the thymus. So this means that in their development, they don't need FoxP3 expression. And indeed, if you look at the FoxP3 binding region, it was published by Sasha Rudinsky's group in Sloan Kettering, and then compared with our finding with DNA Treg-specific DNA demethylated regions, TSTR, completely different, no overlapping. And if you look at the gene expression around the demethylated region, so within 20 kilobases, if the genes are around here, they may be dependent on, somehow influenced by TSTR. And we found that almost all of them, they are highly upregulated, meaning that TSTR is a kind of enhancer. On the other hand, FoxP3 binding sites, and then those genes are affected by FoxP3 expression, FoxP3 binding, they are depressed when they are stimulated. So this means FoxP3, a transcription factor, this is a kind of depressor. So this is a current our understanding of how the Tregs are, their function maintained, how they develop. So with TCR stimulation, the chromatin remodeling happens, and importantly, Treg-specific super enhancer is activated, which controls the FoxP3 expression and other genes, and also epigenomic changes. And the FoxP3 expression and epigenome, Treg-specific epigenome together, now we have a functional, full functional Treg. That is important for clinical use of regulatory T cells. And also for defining regulatory T cells, how we can define them. So I told you that first we showed that CD25 is a good marker, but it is also expressed by activated T cells, then FoxP3. But we know that FoxP3 positive cells, some of them are not so stable, soon losing the suppressive activity. And perhaps most important definition is pattern of Treg-type epigenome and also transcriptome, which must be very robust. And then these cells constitute the cell lineage from the thymus to the periphery. Now let me a bit talk about human Treg. So here this is peripheral blood of normal humans, and you see that CD25 and FoxP3 is a good correlation. But then we know that FoxP3 positive cells, they contain not only regulatory T cells, but also non-suppressive FoxP3 positive cells. So how we can dissect them? And so by using various markers, we separated the FoxP3 positive cells into 3 populations, 1, 2, 3. So if you look at the epigenome status, this population here, we call fraction 2, the mature, highly differentiated Treg. They show Treg-type epigenomic changes. But here, FoxP3 low cells, they are similar to naive T cells. And this here they are called naive T cells. Once they are stimulated, they differentiate into fraction 2. And it has a good correlation with a function. This is in vitro suppression assay. Responder cell alone, if you stimulate them by the TCR stimulation, they proliferate. But the co-culture with fraction 1, fraction 2, fraction 1, fraction 2 suppress the proliferation. But the fraction 3, they don't suppress the proliferation, not suppressive. So this is a current understanding of human Treg. The thymus produces the normal conventional T cells and also the Treg, FoxP3 positive cells, naive Treg. Once they are stimulated, they differentiate to fraction 2. They have Treg epigenome, very much proliferative, so radiosensitive. After suppression, majority of them will die by apoptosis. They are expressing CTLA4, and CTLA4 is important for suppressive activity. And other marker, let me tell you the specific marker of this population, CCR4. And also the separation is important in human, look at analyzing the human disease. So here, sarcoidosis and active SLE both show the expansion of regulatory T cells in the peripheral blood, usually around 70% to 20 or 30%. But which population expanded? It's different. In sarcoidosis, fraction 2, highly suppressive one, expanded. So sometimes you see in sarcoidosis, the cell-mediated immunity is suppressed. Even spirulina reaction, positive one, become negative. Perhaps this is because the Treg suppressive one, they have expanded. And active SLE, non-suppressive fraction 3, they have expanded and producing lots of the pro-inflammatory cytokines. Now let me move on to the cancer immunity. So in the blood, you have 3 populations. But then, interestingly, in tumor tissues, effector Treg, highly suppressive population, specifically, selectively expanded. So this is the case with ovarian cancer, usually E-Treg, effector Treg 2%. Now they become 30%, and no other population. Colon cancer, 20%, malignant melanoma, nearly 40%. So in the tumor environment, lots of the Treg abundant, and they are suppressing the effective tumor immunity. So idea is how we can remove regulatory T cells, especially in tumor tissue. So what is the specific marker for effector Treg? So CTLA4 is highly expressed by effector Treg, and also chemokine receptor, CCR4. So CCR4 is the ligand is CCL22. It's a tumor-infiltrating macrophages. They produce lots of abundantly produce CCL22. And then regulatory T cells are expressing CCR4. They are recruited to the tumor tissue and suppress effective anti-tumor immunity. And importantly, fraction 2, they express CCR4, but the fraction 1, they don't. So here, the fraction 2, they specifically express CCR4. So CCR4 is a good target. So this is a cell-depleting anti-CCR4 treatment of the patients, lung cancer and esophageal cancer. So before treatment, you see fraction 2 cells, here esophageal cancer around 5%. And after 4 times of anti-CCR4 treatment, now fraction 2 cells gone, but fraction 1 it is preserved here and here. And this esophageal cancer patient, he responded very well. So before treatment, he had a metastasis here. Now after treatment, it became a scar, and this metastatic region now disappeared. Now in Japan, we have a large cohort clinical trials to evaluate the efficacy of anti-CCR4 treatment. And another example is the leukemia. It is called adult T-cell leukemia lymphoma. This is endemic in Japan. This is due to HTLV1 infection, retroviral infection. And worldwide, around 20 million people are infected. And 5% of them eventually develop this leukemia, ATL. This is a very aggressive leukemia. The survival time after diagnosis is with less than 1 year. And these leukemic cells express FoxP3, CD25, CTLA4, and CCR4, and very immunosuppressive. Perhaps they are kind of malignant transformation of regulatory T cells. And also, 90% of ATLL cells express an CT antigen, cancer tissue antigen, like an NY-ESO1. And so it's a good the target for cancer vaccination. So this is a treatment for the before treatment, 90–80% there are leukemic cells expressing FoxP3. And after treatment, leukemic cells reduced in number, and also regulatory T cells are all depleted. And this particular patient, before treatment, this is NY-ESO1 specific CD8 reactive CD8 T cells by tetramer staining. So 0.14% they are specific for this NY-ESO1 antigen. And then after treatment, now they have expanded to 3%. And if you stimulate them, these cells with autologous the leukemic cells, before treatment, no cytokine production. But then after treatment, now they produce a lot of cytokines. Actually, this patient now he has survived more than 3 years now. So let me summarize my talk. So for immune enhancement, namely the removing Treg, reducing Treg number or their function, now you can enhance the immune responses, like, for example, to evoke tumor immunity or microbial immunity. To do that, we must reduce the fraction 2 effector Treg or block the differentiation from fraction 1 to fraction 2. So anti-CCR4 is a good reagent to deplete effector Treg. And also, as already Dr. Cantley touched on, the PI3 kinase delta inhibitor, somehow it affects the Treg the number or differentiation and enhance tumor immunity. So we are working on any small molecules which is effective in specifically, selectively reducing regulatory T cells in the cancer tissue. On the other hand, to strengthen or to suppress immune responses, for example, for treatment of autoimmune disease or allergy or inflammatory bowel disease, we must expand the effector Treg, hopefully in an antigen-specific fashion, or facilitate the differentiation of Treg from naive Treg to effector Treg. Or it is easy to make a conventional antigen-specific T cells. But and also we can induce FoxP3 expression. But what is difficult is to install Treg-type epigenomic changes. If we can do this, then the more easily and more effectively we can prepare antigen-specific regulatory T cells, stable ones, and we can use them for treatment of various immunological diseases or suppress organ rejection in organ transplantation settings. So that is what we have been conducting our research from neonatal thymectomy in mice to the recent clinical application of regulatory T cells. So many students and the postdocs are all involved in this project. Their contribution is very enormous, and it is also a collaboration with many clinicians. Thank you very much for your attention.